at the Dutch Medicines Evaluation Board and chair of the Methodology Working Party of the EMA.
Khadija Rantel, Senior Statistical Assessor at the MHRA in the UK.
My name is Tommi Nurminen from FIMEA, which is Finnish Medicines Agency. I'm a biostatistician.
Antero Kallio, also from the Finnish Medicines Agency. I'm a clinical assessor.
Lukas Aguirre, I work at Paul-Ehrlich Institute, which is located in Germany, as a statistician, and quite recently, I'm also alternate member at the Scientific Advice Working Party.
I'm Wolfgang Jacquet. I'm a biostatistician with a Belgian agency.
Hi everyone, I'm Lorenzo Hess. I'm at the Swissmedic biostatistician. Swissmedic is the Swiss regulator, and I'm also a member of the E20 expert working group.
Great, you can keep that mic there for questions. So please, Fredrik.
Hello everyone, I hope you can hear me well. I'm Fredrik Örn from Janssen Pharmaceutical Companies of Johnson & Johnson. So I will start here by introducing my question to the regulators. So I have a relatively straightforward setting, I would say, in a way where we're trying to get a combination drug approved. So we have 4 arms, straight randomization. We have a control arm. We've got 2 monotherapies, M1 and M2. And then we've got an arm combining these two, M1, 2. I denote that. I'm thinking here that none of M1, M1, 2 is currently approved. And the main interest here is really in showing that M1, 2 is superior to the control arm C. Though we could also have interest in the monotherapy arms, M1 and M2, but the main interest is in M1, 2. And typically, at least to my experience, what it says in guidelines is that just showing then that M1, 2 is superior to the control arm would not quite be suffice for regulatory approval on its own. You might need to show some additional properties of efficacy of M1, 2 in relation to, for example, the monotherapy treatments. So we may be interested in showing that M1, 2 is superior to M1 alone or M2 alone. So sort of do both these components add efficacy in this context? And what should the criteria for that then be? Do we also have some criteria for did M1 and M2 turn out to be efficacious in this particular trial, or did that end up to be sort of they performed poorly in this trial, and for that reason those comparisons are sort of easier to show in a sense? So overall then, I'm kind of interested in, for example, multiplicity control procedure. We can see here we have a lot of comparisons of interest. Typically, we might start by testing M1, 2 versus the control. That could be relatively straightforward, but what about all the other potential assessments of interest here? Do we need to kind of strongly control family-wise error rate for all these assessments? Could we think of some other criteria, perhaps nominal significance, so somewhat less rigorous assessments could be possible? And of course, in such a situation, I think it's critical to engage upfront with health authorities, because these could be more or less well-powered, these assessments. How should we set up this trial? What is critical to show? There are also lots of correlated assessments here, so could that be built in somehow? Then, taking this one step further, we consider that might have interim analysis. So think of just the group sequential setting. We test all of these over time. That can kind of be an advantage, I think, because if these are more or less well-powered, we can sort of test them at different time points where there is an adequate amount of data to assess these hypotheses. However, I think you can also see that introduces further complexity. Even if we have a single regulatory approved endpoint, there are a lot of tests here. Now these are also tested over time. On top of this, I think there could be operational challenges in that if you cross, let's say, M1/2 versus C at an interim for efficacy, is it possible to sort of wait for more data for the other assessments? Or will those be hard to interpret if you already crossed the boundary? Does that kind of affect operational aspects for the remaining of the trial? And does that mean in practice, really, we need, if we now are very rigorous about having all of these assessments in this trial, does that mean in practice, we really have to cross the boundary for all of them at one interim? It's hard to sort of wait for data for the other assessments to assess them at a later interim, or could that actually be possible that we cross the interim boundary at an interim for M1/2 versus C and wait for sort of more data for the other assessments? And then finally, I mean, looking at it from a slightly different angle, suppose now we can assess this M1/2 versus C, either at an interim or at a final analysis. So the main comparison has been shown. Could there be complementary means to assess the other hypothesis? For example, consider perhaps other endpoints of the same trial, perhaps Bayesian approaches incorporating historical or external data, or other maybe more model-based approaches based on the mechanism of action. An additional consideration is then that sometimes we have the 2 trials rule. We need to show this in 2 trials. That, of course, increases complexity further if we now have to assess very rigorously all of these assessments in 2 trials. Well, could we think about 1 trial with more stringent alpha, for example, or other ideas?
Okay, thank you. Okay, so there was a lot of questions. Let's start with the beginning. So whether the contribution of the individual components needs to be shown and how. Kit, please. You have under the mic for Kit.
It's on now. Before we dive into the details, I'm inclined to give a general reflection and start with another role I have. In the Netherlands, there's a program funded by the government to invest in large clinical trials to optimize drug therapy, which all happen for drugs that are already on the market. And there are lots of good proposals and lots of research questions that are quite relevant to address after licensing. But I also see proposals which are aimed at getting rid of treatments that actually have a weak evidence base, and there is doubt whether it's actually adding something. And those proposals frustrate me because there will be millions of euros invested in a trial, and usually that will become a non-inferiority trial, to get rid of a treatment that should actually not be there because the evidence base is too weak. So I would like to make a general point before we dive into details. This is quite valid. This is a combination. So you better be sure it's the combination you need. So I would kind of promote before we dive into details with this in mind, to have a fairly strong standard of evidence before we move into the details on how to do that efficiently. Because you would rather not go in the trajectory that I just sketched. And luckily, most of those trials now proposed are very relevant, but I do see some of those proposals, and I think that's kind of frustrating because that could have been avoided and avoided costs. So that's a general point I would like to make before we dive into details on how you do that efficiently.
Yeah, Florian, please.
Yeah, thanks. A lot of questions, and I think we could fill a whole session with those. So it's hard to answer all of them. And I think I completely agree with Kit that the standard has to be high, especially with combination drugs, so that we don't add anything that doesn't add additional benefit. And so from that perspective, sorry. From that perspective, I think at least demonstration that the combination is better than each of its component is a very high value goal. And so family-wise error control is our current standard of evidence that we require, and I think that sticks. Comparison to another standard of care like control, I mean, that may depend on the situation. What do we know about each drug and how well established is the efficacy for each of these drugs? If you need it, then also maybe to show assay sensitivity or something, then again, it would be part of the overall confirmatory strategy. Of course, in such a situation, you have logical relations. So you are making comparisons that something is better than something else. And if that's better than something else, you can have some implication that can help you with your procedure that you use then. Yeah, and then making it more complicated with group sequential or adaptive designs. I mean, I don't see why you should get a discount by doing a more flexible design on type 1 error control. And there, of course, as you said, you have some operational challenges if you can demonstrate only some of your objectives early and others. But again, I mean, you could structure probably your strategy in a way that you first go for the most important ones and then maybe can stop some of the arms in your 4-arm trial, leaving maybe only the single components and the control just to have the demonstration of SSC sensitivity at a later time point.
Benjamin, please.
I'll start with the last point. I think the trial planning and when you conduct an interim analysis, this is really a crucial exercise you need to think about in the beginning when you plan the trial. So we recently published a manuscript where we tried to address this, and I think this is really important, regardless of the aim of the trial, to really consider when to conduct an interim analysis and with what purpose. Do you have sufficient data at hand to support filing, or is it just a futility analysis? Well, then that's of course another issue. But I think you should really carefully plan the timing of the interim analysis. This is one point. And with respect to the question of what to compare with what, I would want to point to there's a guideline on the clinical evaluation of fixed medicinal products. I think that's a fixed combination of medicinal products, something like that. And this guideline only requests that you compare your combination arm to the single components. So it's a 3-arm trial, which is proposed in that guideline. And I think we can ask ourselves, what is the reason behind that? And for me, the reason is we want to really show, and this is what Kit has said and what Florian also said, that we want to show that each component has a substantial contribution to the overall effect. This is the general rule, and the guideline also outlines specific situations where you don't need to show the contribution over a specific component when you don't expect any contribution of the single component a priori. So for example, it might just be a treatment enhancer and you know that from the beginning. So you would not even need to have that arm in the trial in such a situation, but then this would require upfront discussions or a good justification. And with respect to the control arm, I would not think that you necessarily need to show that your monotherapy is superior to your control. So because I would assume that, I mean, the most important thing is that your combination therapy is superior to the monotherapies and to the control, depending on the situation. When you would expect any contribution of the single components, then it would maybe even be sufficient to only have the 3-arm trial and to only show that you're superior to these monotherapy arms. But this is really also then dependent on the situation and indication and drug you have at hand. But I think the most important comparison, this is what I want to say, is the comparison of the combination therapy to the monotherapy arms, or if available, also to the control arm.
Yeah, please.
I'm thinking in your example with a 3-arm trial, though, that's in a situation where M1 and M2 are approved, presumably, because if they're not approved, how do you then you could compare in theory to something that's worse than placebo in theory. So
Well, that's correct. That's what I meant but this is the guideline, and I'm not sure whether the guideline says that these are approved. I think they don't consider the situation. But of course, it would be helpful. And I think so the control arm is really helpful to try to understand, is there any effect, or is it worse than placebo? So do you do harm to patients? But this also then you would have data which helps you to understand whether you would do any harm to the patients with this mono component. And hopefully you could also rule out this by external data. So that's what I say. So the main component is the contribution of the combination over the monotherapies. But of course, you're right. We would need to exclude that you do harm by the mono components, and that you compare to something which is inferior than placebo, which I'm having troubles to see. It would be a strange drug, and I hope you don't develop these drugs. But yeah, of course.
Then, I mean, if I can, I mean, you could imagine also that it's another situation that you have a really high hope that the combo is really efficacious, but you're not obvious that the benefit risk is better than for the monotherapies. And you might not know that up front. So you might want to, from a sponsor side, perhaps plan for all of these assessments, and maybe winning on one of the monotherapy could be really important if that has better benefit risk. And from that perspective, then it could be more convincing if that also has been shown with control of family-wise error rate for that assessment as well.
So this is the short topic session. Maybe we can evolve it
Yeah.
also in a short answer session. So
For me, the most important starting point is really the rationale for the combination. What is known about the individual monotherapies, and of course, in terms of showing efficacy, if you are proposing the monotherapy, then obviously the statistical significance for the combination is the key. And anything else is possible, but it depends on what you have pre-specified. So it's very important that you think about what testing you're going to apply during your study, and then pre-specify that. What is not acceptable is to come up with hypothesis testing once you have looked at the data. So pre-specification is key. So, and I stopped there. So hopefully it was a short answer.
I think we can take one more short comment and then we need to move on.
Very short. I try to combine everything a bit. So you're interested actually, and you hope, and you think that your combination will be extremely efficacious. So then you can say, I'm going first because you said a time element. I do a hierarchical approach, and I actually go for first testing the combination with placebo. I know that if that is successful, I still need, according to the guidance, to go further. But I presume that the monotherapies are going to do something. So that means that I'm actually inclined to say, I'm going to test at a later stage when that's more mature. Okay, and then you go for tests later on. That gives you hierarchy, completely acceptable. Some dynamic elements, completely acceptable if you actually can design that well. So you need enough information. The problem there is, of course, that what is the risk? That is that you fail the first, and then it goes wrong, but you still go on with the trial, and then you say, oh, please accept something, because there is something. And that is actually something that pops up, and it is completely unacceptable. And we should think about those cases, and what are we going to do then? And that means normally new trials, point period. But that seems to be too costly, too time-consuming. So there is the real issue, correct, but risky business.
Very quickly, Rans, and then we can.
I very much agree with what he said, and also what Kit said. And if you think about it, none of the drugs is approved. So we don't know anything about the dose finding and the added toxicity of the single drugs and then of both drugs. We don't know how the sequential treatment would be of first M1 and then M2 or vice versa. So I think this trial is very, very, very ambitious, and we jumped on the statistical things, but I think we have to do one step back and ask ourselves which are the real questions here.
So maybe phase 2 development could be an option. Okay, thank you. We can continue this discussion, I think, over the lunch. And please, Betty, Next question. This question is for sure shorter, but not necessarily easier to answer. Please.
Thank you. So I'm Betty Malloy from Novartis, and I'm presenting this question on behalf also of my colleague Evgeny. It's a very short question, so basically we want to prompt a little bit of discussion about what it takes to get real-world evidence into a drug label. And so we have a scenario where we had some scientific advice. Sorry, I'm just going to wait for...
You can just continue if you.
You can just go. Yeah, so we had some scientific advice where we discussed beforehand with the EMA on how we would contextualize the data from a single arm study. And we had a plan to do this with indirect comparisons to patient level external control data. The feedback we got that this was considered useful in helping to understand the efficacy of the drug in the current therapeutic landscape. We move on 18 months, we've had a positive readout, we've submitted our data, and now we propose to include the results of this indirect comparison in the SMPC. We want to make sure that this comparative data can be of use to clinicians and prescribing physicians in the decision-making process for the treatment with this drug. Especially at present, there's no clearly established treatment. Our therapeutic options are very heterogeneous in this line of therapy. At this point, having been told beforehand that this was useful information, this was very useful to contextualize and stuff, we got told, well, no, you can't put this in the SMPC. So then our question is, why is this real-world evidence useful for EMA, useful for contextualizing the results, but not useful for prescribers? What is the regulatory advice to sponsors for inclusion of real-world evidence into the label, especially when it's pre-discussed, pre-planned, pre-collected? And you know, what can we do then in these situations? What has been your experience, and what is the advice you would give to companies who want to include this sort of information in the SMPC?
So maybe to streamline the answers, so you're especially for now, you were looking for an external comparator arm, right? So it's a comparative situation, which is different maybe than real-world evidence as standalone for a standalone question, right? So we now discuss the comparison. I think here, because it's a short topic session.
Yes, okay.
Yeah. So it has been a comparator arm into a single arm study, I think, in this case, what we are discussing. Maybe Andreas could start. We have the second mic there.
No, okay. But first of all, I have to say, I don't think there's a contradiction between the advice that was given by SORB and that you should not include this information in the SMPC, because you have to see, I mean, contextualization is of course important during benefit risk assessment and for decision making, but the SMPC is actually not the document that should reflect the decision-making process and benefit risk assessment. I mean, it's mainly a description of the characteristics of a specific product. And so this external data is external, so this was not created in relation to the specific product. So I don't see a direct contradiction there. And whether an external comparison can be included in an SMPC, of course, there can be no general advice given. It's always a case-by-case decision, but the general principle that is methodological robustness. And of course, for these single-arm trial comparisons, we know that there are several limitations, and these limitations have to be reflected. And I think the right place for that would be the EPAR, where all these details can be included, but not necessarily the SMPC. So currently, I think our policy would rather be not to include real-world data at all.
This was a very
The SMPC,
regulatory
sorry.
process type of an answer. What is
It
the right
sounded familiar.
place? Yeah. Okay. Hadisha, please, maybe from MHRA point of view.
I agree with what's been said. I think the issue for us as well would be that when you are trying to present information on the SMPC, you want robust and helpful information, but we know with the real world data, when we do the benefit risk assessment, we have that further insight into the quality of the evidence, which obviously these level of details we cannot present in the SMPC. So if you just present that information as it is, without the context about the robustness of that evidence, etc., then this could be a source of misunderstanding and misuse. So I think for that reason, it's not the place for it. That's my position. Thank you.
I mean, we have seen real world data make it into other labels and stuff. So I wonder if you've got experience where you have considered that something can go into a label, or what the difference might be.
I have no experience where we had actually used that evidence from real world data in the SMPC, but again, I think there may be a case. So say it's
maybe
never
Benjamin
say,
can give us
but
the case study.
I have not come across one personally.
I've come across one and I was also involved personally, unfortunately. I have to admit.
Fortunately or unfortunately?
Unfortunately. And we had internal discussions on the role of real-world data, and I would want to refrain from using real-world evidence here because it already implies that we have evidence. But first of all, it's just data which is collected, and the amount of evidence which is generated by this data is questionable, and this is part of the discussion. Internal discussions then led me to understand that this is not something we should place in the label. We had done this for CAR T cells, for example, in the past, where we described also the historic controls. But it's not about the drug. So usually the SMPC should describe drug effects, as Andreas has already elaborated, and it's not about the drug, and it's also not related to the comparison because it's just contextualization. So it's even less. And as said, it's such a complex discussion. We cannot have this in the SMPC. So the right place for this would be the European Public Assessment Report from our end, and we try to reflect this there. And for companies, of course, you can also publish your results in journals, and this is what you do. And this would be the right place to give contextualization. But in the end, it shouldn't be part of the label. And yes, we did this in the past, but there's no, we cannot always look back, and we did do wrong in the past, and this does not make it good in the future.
Kit, please. Oh, you have some stuff? Yeah.
Yeah, there's one question, just it's more for Betty, I guess. Can you clarify whether the data on this external comparator was pivotal for the benefit risk, or was it just supportive? So I guess maybe just to give a bit of context.
I think it would have been considered supportive. It did make it into the assessment report and was considered to have been supportive in contextualizing the results. Yeah.
Take a slightly different view and have a positive note there, possibly. And it's also looking at the position that the SMPC has in guiding treatment or treatment guidelines. You would be surprised how little it is sometimes used in developing guidelines that physicians subsequently at national level follow, because they will tap in not only the SMPC, but actually will give priority on everything that's published. So I would, my positive note would be here, just because it's not in the SMPC, doesn't mean it goes unnoticed for those that you actually are targeting. But if it's in papers and in context and together with all kinds of other external data that you have not made part of your submission, is out there. So I see the point. I think the points why it's not there are clearly raised, but I would also promote, do not worry immediately. If we're transparent and we publish, the external data will also guide treatment guidelines.
You content with the responses you get?
I think they've been comprehensive. Thank
Good.
you.
Great. Thank you. So next we have Patrick. Patrick, please.
Thank you, Elena. Hope you can hear me well.
Yes.
When Kaspar's putting up the slides, briefly introducing myself, my name is Patrick Schlummer. I'm a principal statistician working at Bayer in Germany. And here today, I would like to present an example of hierarchical composite endpoints. Thank you. Specifically tailored towards chronic kidney disease trials. This is joint work together with my colleague Christoph Tasto at Bayer, as well as Henrik Thomsen and Henrik Raven at Novo Nordisk. The usual disclaimer has also been put on this slide, so my freedom of speech is ensured, which I like. Starting with a brief introduction on chronic kidney disease trials, where traditionally in the past and also still today, composite time to event endpoints have traditionally been used as the primary endpoint, consisting of the following components. Usually you have one component which represents a certain relative decline from baseline, which needs to be sustained, meaning that it should be confirmed by a second measurement at least 4 weeks after the initial decline. Typically, you have thresholds like 57% or 50 or 40%. GFR here stands for glomerular filtration rate, which essentially reflects how well the kidney is functioning, so the higher the better. Another component typically included is a sustained decline below 15 ml per minute, which is typically then resulting in further actions like dialysis or transplantation, also referred to as end-stage kidney disease. And last but not least, some form of mortality is included, usually disease-specific mortality like renal or cardiovascular death. More recently, GFR slope has been suggested and also seems to be increasingly accepted in CKD trials. So the rate of change in GFR over time assessed continuously. Some of you may have seen the recent draft qualification opinion that has been published on the EMA site on this topic last Wednesday. So perfect timing for this meeting here today. This obviously leads to more efficient analysis, smaller trials eventually. However, handling of intercurrent events like death and end-stage kidney disease is not relatively straightforward, as subsequent eGFR measurements are either non-existent or not interpretable. So this is the combination essentially of the traditional and the modern approach by means of a hierarchical composite endpoint, here abbreviated with HCE. On the right-hand side, you see a graphical illustration, just an oversimplified example of one patient that starts with a GFR of 40, and all the different components are included here visually, starting with a triangle, the green triangle representing GFR slope. Then the patient reaches 40, 50, and 57% relative decline from baseline. Then the GFR falls below 15 ml per minute following the patient went on to dialysis or receives a transplantation, meaning ESKD, and then the patient dies, which obviously could also happen at an earlier point in time, but just for simplicity reason, we've put it in here. And you see almost all the information comes from the same disease-specific domain based on GFR, and the ordering of severity, I would say, is relatively straightforward. We've listed it here on the left-hand side. We start with all-cause mortality, then we have end-stage kidney disease, the drop below 15 ml, and then the different relative thresholds of 57, 50, and 40%. And last but not least, in contrast to the traditional composite endpoint, we enrich the endpoint by the continuous total GFR slope. So just picking out the two most relevant attributes of the underlying estimate, we have the variable, which would be the most severe event out of those 7. And for the time to event components, we also consider the time to the event. And if none of those events occur within a certain time period, t, the total GFR slope at that time point t will be considered. And the population level summary is then the odds that a specific patient would do better on, would have a better outcome assigned to the intervention than when assigned to the control. So that could, for instance, be estimated with a win odds, which might be familiar to some of you. So coming now to the questions. Also, again, in the light of the recent draft qualification opinion on GFR slope, which speaks rather positively about the use in CKD trials, we would be interested to hear the general views of the regulators on the application of such an hierarchical composite endpoint in CKD trials. Could you see it as a primary endpoint, maybe rather as a secondary or exploratory endpoint? And the second question would refer to the interpretation of such an endpoint. Would you see it A, as a holistic assessment of kidney disease progression? Would you rather see it B, as an assessment of GFR slope, which is kind of accounting for certain intercurrent events in this regard here with a composite strategy? Or, of course, I've added C because I know that you regulators are typically very creative. So I leave it open to you. Happy to hear your views.
Okay, thank you. It's very different kind of question from the previous two. Wolfgang, please.
Okay, I welcome this very much because all the GFR slope in the first slide, you missed something. That is, it seems to be reliable, it is not. You've got the first phase where actually there's a lot going on. You've got decline, decline going up, and then you've got the slope. But how long is that first phase going to be? It's dose dependent, it's product dependent. And if it is taken into account in calculating the slope, it's actually geometry. I've got a slope there and I'm going to cross a threshold and I do this. And a small error here gives you an enormous swipe over there. So actually, there should be pro and cons to using the slope. The slope is information, but it has to be treated more carefully. So in general, I think this might be something good if well designed. And a role for the slope, but well designed. General remark, that is.
Lucas, please.
Yeah, thanks. I may have slightly different opinion on a few aspects. It's I think it's a like good way to merge different things in statistics. But as usual, the difficulty lies in linking statistics and benefit risk. And I would find it rather challenging to do a benefit risk on the summary measure, the odds ratio for this endpoint. So that said, I guess it can add value for any type of application, but it's probably very challenging to base the key interpretation on this. And that is partly also because the different events that are included here are of different relevance, obviously, like 40% and 50% decline are very different from death. So this would be very challenging, and you would need a lot more information to make sense out of it. So that's just a remark.
If I may ask.
I think now you turned it off.
If I may ask a follow-up question, I know there's a lot of controversy on the interpretation of the actual point estimate of such an endpoint like the win odds or the win ratio. Could you rather then accept seeing this as some form of a gatekeeper to declare whether a trial would be successful or not, followed by an detailed assessment of the individual components, which is anyhow always done for composite endpoints?
Well, in a way, yes, but I guess it depends on the case. Sorry.
It's right, you know, to have a conversation.
Yeah. So you would obviously need further information. So in principle, yes, it could be a gatekeeper. We use the approach to have a gatekeeper and then afterwards a benefit risk assessment in other situations as well. But it's very challenging probably here to define what sort of additional data you need to be able to make a benefit risk assessment. And that is typically defined by the primary endpoint. So if that's not the case here, then it might be difficult.
Tommy?
Yeah, maybe just to lower the bar for any potential comments to follow. I have no experience in this kind of disease, and I struggle to understand the logic of why it's time to the most severe of the first 6 components. So, I mean, someone dies at time t, and someone has, let's say, 40% reduction at an earlier time. So doesn't it mean that the one who had 40% reduction in kidney function at an earlier time actually had a kind of worse outcome, although the other guy died? Okay, please correct me.
Of course, I forgot to turn it on again. It's always the most severe outcome that counts for the comparison. But if a patient dies and the other patient did not die, then the patient that didn't die wins against the other patient. So it's kind of some form of ranking that will be done.
But is the time then long? If somebody has all these events, but also eventually dies, gets a long time.
Mm-hmm.
Okay.
But the death will then be considered, not the other events prior.
But then somebody who had one of these events but didn't die has a shorter time for that event, that they had the same event that they both had. The other guy died and the other guy didn't.
Mm-hmm.
But they had both the number 4. For the other one, the number 4 counts, and for the other one, it doesn't.
No, it's only the information that the patient didn't die within, let's say we consider 3 years. One patient didn't died after 2 years and the other patient just was still alive at 3 years. Then it's the win for the patient that still survived until 3 years. And all the other information is considered irrelevant for that specific comparison. But maybe you want to comment on that.
Yeah, I think I know we know these approaches, they are also used in other areas, but I think firstly, of course, the question is, do we really want to split this testing and estimation with, do we want to go forward with this gatekeeping approach? Because then we only know it does something, but we don't know what it does. And what you just discussed also with this ranking of these events, it's really very difficult also when we talk about intercurrent and so missing data replacement. Actually, it's a nightmare if you don't want, if you want to do some kind of a missing data replacement, because you would need to make it in a multivariate way. So I don't know how this could be possible.
May I comment? I mean, I personally don't see it as too complicated, because just imagine a patient simply does not have any GFR measurements after 2 years. We would have to do some form of imputation on that one level and could infer almost all of the other components from that. So it's not too multidimensional, I would say.
Okay, yeah, when you're that optimistic. And okay, but maybe another comment on this question, what it actually is. I think this brings us to this key question: what do we actually estimate? So when we asked on this last slide, what is the estimate that we actually target? I think the problem with this holistic assessment, the problem is this endpoint can mean a completely different thing depending on the follow-up and the patient populations that you study. So I would not go for this holistic interpretation. And the assessment of eGFR slope accounting for intercurrent events, I think this is kind of some kind of a misinterpretation of the composite strategy, because from the estimate framework, because the composite strategy means that all events that you include in the component have to be considered and have a clinical meaning. So these are not intercurrent events anymore. These are actual events of interest. So these are part of the endpoint. So I would go for something else, but actually I don't know what else it is.
Could I make a brief comment? Sorry, just from a HTA perspective on this, and I think this point has been made from the regulatory side as well. But this is a very challenging. This population level summary is a very challenging effect to interpret in terms of meaningful outcomes on patients. And I think if a drug approved on the basis of this endpoint, we're then going to HTA, I think we'd have an extremely difficult time actually quantifying those benefits. Either in a system where you assess clinical benefit alone, or indeed when you try and turn these outcomes into a cost effectiveness model as well, which would also have to take that into account, because ultimately, this odds ratio, of a better outcome incorporates, improvements of hugely different levels of significance, and it's very hard to interpret in terms of, the meaningful sort of, meaningful effect on patient relevant outcomes, and I think that would be hard for us from HTA, and from what I hear, I guess from the regulatory side, there are maybe similar concerns.
There's one aspect that is problem with kidney disease and why we use slope. I think everybody knows that slope is not optimal, but it is because those trials are extremely long if you don't. Now, my question is, this approach will not shorten, so I welcome something better than the slope. Certainly, in view of the beginning phase, we need to devise something that performs better. But the question is this going to be it in view of the design, multiple problems?
I just wanted to make one addition. I was rather negative before, but I think this, I really like the question. It's extremely challenging, and it makes you think about under which circumstances could we accept that and interpret that. And I think that there are scenarios, like if you are on one edge of the distribution, then it's probably easier. If there are no deaths, then you could just take slope. And if there are many deaths, then you could just take mortality as an endpoint. So the problem is that we are somewhere in between, and maybe this tells you something about the population and the effects that might be addressed by such an approach. So it's really a challenging question and a good one. I like it.
Please.
Very brief addition. I think that the problem is, aside from the difficulties in interpreting the primary endpoint you propose, that you might not have sufficient data to robustly estimate the contribution of the composites, because this is the reason why you use this endpoint in the first place, I would assume. And this makes it challenging overall. So this is my take. Not saying it's not possible, but it makes it challenging.
I agree that sample size calculation might not go the traditional route for such an endpoint. Maybe there would be requirements for certain number of events that are part of that endpoint. So yeah, it's a point well taken. Thanks.
Thank you for the good question and good discussion. Not easy. And Simon is next. Change the slides. So this is a nice title. What's the rule for the pool?
Yeah, that's kind of the overarching question. Yes, so I'm Simon Wandel from Novartis, a statistician as well. I hope my voice survives. Yeah, we wanted to ask something specific about a situation which doesn't occur so frequently. There are some disease areas or circumstances where the two-study convention is not commonly enforced. I think in oncology is a very classical example, cardiovascular diseases where I'm working in, and also rare diseases. But occasionally you may have a situation where two pivotal trials are conducted, but these may have a substantial lag between their anticipated readout dates, or it can be several years, for example. They may enroll populations that differ to some extent, including geographic footprint, maybe some variations in inclusion-exclusion criteria, so on and so forth. But let's assume it's not to an extent that you would say these are really different populations, right? But, that you say maybe the inclusion-exclusion is not exactly the same. And they may also embed a different testing strategy, for example, the order of endpoints between, which they test can be different. Now, for endpoints with low power, for example, mortality, pooling data across two studies is, of course, highly attractive. And the key questions for us is, of course, what does it mean with regards to type 1 error control? So let me see if I can move that. Okay, so here are some selected perspectives, let's say, that we are aware of. There was a draft version of an FDA guidance. The text I highlight on the right-hand side was removed from the final guidance, but it somehow, was specific about cardiovascular mortality there and about type 1 error control. So this topic somehow had been, part of the discussion on the FDA side, it seemed. There is another FDA guidance with more general considerations. There is the EMA point to consider on application with first meta-analysis or second one pivotal study, which has some interesting material there. And it goes exactly into this problem, right? If you have an endpoint that you may not be able to power in the individual study. And there is also a statistical framework that Frank Bretz and colleagues have published. The limitation of that framework in a way is that it really works well if you have two trials basically exactly the same and they go in parallel, because you have like a gatekeeping of the same endpoint that you need to hit in both studies first, and then you can go to the joint testing in the pool. So the specific questions we wanted to ask today is consider a pre-specified secondary endpoint that is not statistically significant in either study since the power is low. And it is usually logistically difficult to power this endpoint in an individual study. For example, cardiovascular death in the cardiovascular area, where it would take you forever, right, to achieve a number of events that you would say, I'm happy with from a power perspective. So our questions are, would the pooled data to be considered as robust and strong evidence of the treatment effectiveness for this endpoint, potentially resulting in a labeling statement or SMPC statement if consistent positive trends are observed from both studies and statistical significance is reached in the pooled data at the nominal level of significance, okay? And would it be recommended to have a separate protocol for the meta-analysis as suggested in the guidance, or would with a protocol-specific alpha, or should a meta-analysis be included in one or both of the study protocols, which would be an alternative with type 1 allocation from within the study? Now, before I let you answer, just let me preempt one answer. Yes, of course, these things will be published, but the question is really about the regulatory viewpoint here. And of course, the goal would be to have a discussion if that could be included, let's say, in a label in general.
Thank you. It's a good general question, but specific sub-questions. Kit.
Yeah, I was kind of prepared to pick the mic because this was actually a topic of a fairly interesting session last week at CEN 2023. So for those of you who were there, you will not hear a lot of new insights from me. I will try to summarize briefer than I did there. I think one point, which is kind of made in between, occasionally there are two trials. I kind of want to also use the opportunity that we actually might be, let's say, ignoring a bit the importance of replication for progression in science. And luckily, it's not occasionally, but it's dropping. So the amount of applications we have where there is actually replication in any way, as it doesn't need to be replication of two exactly the same trials, it is reducing. And I think that's an overall scientific concern. So I would not go too quickly to embracing everything with one trial. So that's a general remark. Moving down to the question on endpoints that are difficult to achieve. Strong evidence within a single trial. I think you're right to refer to the one pivotal meta-analysis guidance that actually precisely opens that opportunity. That of course, if you conduct multiple trials, not every question can be reliably answered within a trial, and there is opening from a meta-analytical perspective to answer some of the questions across trials. So I think that's generally supported, and it's in line with guidance. The question is, of course, when does that become robust evidence? I think that's the key underlying. And then a couple of thoughts there. I think there are pre-specification, whether it's in a separate protocol, before the studies start, including also sample size considerations on that secondary endpoint and reassurance that you will be able to do that across the trials that you plan. I think that will be a very important component. So not an after-the-fact meta-analysis, but a pre-planned and including up to including sample size considerations. Not we have 2 trials and this is what we can do, but can you actually convince that those 2 trials are actually enough? Or is it still too small? So that's one. We've also discussed some of the scenarios there in terms of error control. I will not call it type 1 error control because I think there we need some discussion. There are lots of schemes there. And my general principle would be, I think I would probably choose schemes where you go into those secondary questions only if you have established efficacy across the trials. So you would do that in a so you can devise all kinds of fancy systems how to distribute errors in difficult scenarios. But my principle would be, let's have this discussion on secondary endpoints only after the 2 trials or the 3 trials that I want to incorporate in that question actually support efficacy. And then only then that second question, and that can be translated in an error scheme. I'm not sure whether we want submission-wide error rate as a concept there. It's not by coincidence that it's not in the one pivotal meta-analysis guidance. You will not find alpha square there as a landmark, and that's not by accident. That's because I think we should probably have some discussion on what we consider error levels across trials and errors that we see as type 1 error belong to one experiment. And I think that's a more general discussion which will also become relevant in more complex trials. So those are my first considerations and a very brief summary of what I kind of shared last week as well.
Anybody else wants to comment? Benjamin?
I can briefly add to what Kit has said. I can fully agree with everything. And I think what is important that we should avoid going in the opposite direction. So the trials failed on the primary endpoints, but we see incredible strong overall survival or mortality results than in the pooled trial. And then we say, well, we need to. I think we should avoid to have this discussion. So I think this prerequisite to have successful trials is really key to this exercise. If we have established efficacy in the separate trials, and then we look at the pooled meta-analysis, however it is done, I would not think that necessarily we need to have a significance attached to that. The question is if we can, but I think it's much more important whether it's clinically relevant and robust and reliably estimated. So these other statistical and clinical quantities of this estimate are much more important than the question of whether it's significant. So we often include endpoints in the label which were not significant. So consider, for example, in oncology, we would rather tend to have overall survival in the label, regardless of whether it was in the testing strategy or whether it was significant, but it tells you something about the drug. So it's really important for patients. It's a really patient-relevant endpoint, and we want to include it because we think it's important for the prescribers and for the patients to have it in the label. And I would assume it's the same here in this case. So if you have a robust evidence and we can rely on that estimate, then it would not be a question of whether it was significantly tested, but whether it was pre-planned, it's robust, and it's clinically meaningful.
I don't know if somebody would have comment on the fact that I think you showed on the first slide that the 2 studies may also be quite different. And this is something I kind of started to think when I read this question, that I don't know if you want to say something to that first, because now all this, I think the actual questions were quite well answered, but I was thinking that, okay, you have two quite different studies and you want to combine them for the analysis of an endpoint. Combining two quote unquote identical studies is for sure much easier.
Yeah, okay. I mean,
so have we missed something?
No. I
related
think
to
it's a good
this?
point.
Yeah.
And I would say, you know, what I want to avoid is to get into it depends, right? Because of course it depends. You will tell me if I have a study in males and one in females, then maybe pooling is not the best idea if, you know, it's an important prognostic factor, let's say, or whatever, right? So I would say, you know, the situation we're thinking of, yes, it's not exactly the same. But I would say, you know, if you speak to clinicians, we have also a clinical assessor here, right? But which is a very important part, of course, here is that the agreement will probably be, it's in a range that you would say, you know, it's logical to pool because it probably spans a bit the broader group of patients, right? But let's say whatever in oncology would say I'm probably still in first line, right? But
okay.
Or if you're in third line, right, you would say maybe some of these patients have different prior treatments than the others, but I wouldn't expect that this makes a substantial impact of how they would respond to treatment.
Tommy, please.
We have seen little challenging from the audience so far, so maybe.
Yeah.
While waiting for the first question from the audience, I think you had a practical question about where it should be pre-specified. By the way, I think we all agree that pre-specification is key here. But we've had very simple cases of two concurrent kind of replicate trials running at the same time in similar populations. So kind of the easiest possible case where pooled analysis across the two trials have been pre-specified. In my recollection, it's been pre-specified in these parallel protocols without any specific meta-analysis plan for this. But I wouldn't mind wherever it is pre-specified, as long as it is.
Please, Uli.
Maybe just to add a little bit to Simon's question, is when you have a hierarchy of testings in your tests, in your protocol, and in the mid you have then something where you combine the data from both trials to achieve the necessary power. Is that an additional view on your side? Would that be acceptable, or do you think that methodologically starts to make more, to put more questions on, etc.? But say when you have, in each study, you have a hierarchy, and then the third level is basically the combination of both, and you need to be significant there to go to the fourth hierarchy in each trial. Is that acceptable?
The short answer, of course, it depends. But I think some of these scenarios were discussed last week as well, and some of them actually do not comply with my first principle, and that's is efficacy established across the two trials before I even start answering the question, irrespective of where you've put the endpoint in a hierarchy within the trial. So that's what I mean. It depends. You only tell half of the story. The hierarchy needs to be such that it complies with the first principle. I'm looking at two studies that actually support efficacy first before I go there, and not all of the hierarchies comply with that principle.
Dorian, do you want to comment something?
Yeah, I think if you have it in a testing strategy and you control some error, the first question is, which error do you control? I mean, on what level do you control the error for the combined test? And I think there's no convention on that yet. And we have this requirement from KIT. And then, of course, you're in a slightly different situation than what people were talking about, because now you want to have a yes-no inference and no longer like an estimate where you synthesize the evidence from both trials. So in the end, you showed that you have an effect on a certain endpoint. And I think that can be acceptable, but I'm not sure if we can already say if it will be because there are still some open questions on how to control the error and which error to control.
Okay. We have another question from the audience.
Sure. Sure. Can you hear me okay? Just challenging you a bit about the mortality endpoint. So you correctly explain that often always you'll have mortality in the label. But when you say mortality was that endpoint, you wanted to look across 2 trials, and you see a sniff of a benefit, then it's going to be in the label because you just said it was. Now you see more of a sniff, and then all of a sudden you see nominal significance, and now you see maybe even more extreme evidence. But the subtlety's there. It'll always be in the label somewhere, won't it? Because it's mortality. So it's quite I mean, there are many cases, I think, where CHMP have approved or given label indications for mortality just because they saw it, but without any statistical control at all. But anyway, maybe you could explore that angle.
I'll take this one because, of course, I've been fairly black and white in my answer, but also to make the principles clear. This is recognized. And I think actually the more general point I would like to make, somewhere you switch between a hypothesis testing state of mind to actually estimation. And this is for me closer to estimation. What are the effects? Of course, it will only be in the label if the product is approved. So let's say if I stick close to my first principle, we want to be convinced of efficacy. And that hurdle needs to be met. And of course, also that hurdle is not entirely black and white. Suppose I have 2 trials. Hopefully not exactly the same, which I've also seen, because essentially that's one trial split in 2. You have 2 trials, both come up with similar, or at least same ballpark range of treatment effect estimates. One is highly significant, the less so, 0.06. You might still be in a situation that we think this is supporting efficacy and allows a meta-analysis across studies. And if you then have that sniff of evidence of that going in the right direction, it actually strengthens your case a bit across the 2 trials. So that will happen during assessment. And then indeed, let's say mortality will make it to a label with the robust possible estimate of the effect size there. So that's how I think it will be in practice. But I think the underlying principle is that kind of first hurdle. If that was not the main endpoint, of course it's important, and yes, it will make it into the label. And then probably, yeah, for my liking, with the best possible estimate you have of the effects there. And I would kind of think meta-analytic. I think we're using pooling. I'm thinking probably a little bit more meta-analytically, acknowledging the fact that the studies might not be exactly the same and actually be real replications.
Okay, so I think the regulators were maybe a bit more relaxed actually than your proposal. That happens as well.
Sounds like, but there is another comment.
Okay, there is one more comment.
I completely agree with Kate. I think for me, I mean, further testing, if the studies are not positive, it's not really warranties, but where there is obviously some uncertainty about the estimation itself, I think pooling of the data does improve the estimation of the treatment effect. The other aspect also to consider is with regards to the pooling is really to be very clear on whether the 2 studies are actually can be pulled together, and that's something that really must take precedence over, you know, the decision to pool. Are we dealing, obviously thinking about the estimate framework and looking at the different attributes and comparing that between the 2 studies once they have reached their primary efficacy, then obviously that is an important aspect to end up with a treatment effect estimation that is much more informative and can make it to the label.
Thank you. Thanks a lot.
Great. Then we have next Sandra. Another provocative question, I guess, this time. And I'm sorry, David, that you're the only HTA person in the audience. So please, colleagues, help him getting the mic already.
I'm Sandro, I'm working at Roche, but I'm presenting here on behalf of the PSI HTA special interest group. And so the objective is to deliberately broaden the perspective, the discussion a bit, and also bring the
Can you take the
HTA and
mic a bit?
reimbursement decision makers into the debate. So the usual disclaimer, the opinions here are those of the authors, not necessarily those of the companies. In terms of background, so now we want to talk about surrogate endpoints. And as you all know, surrogate endpoints are used a lot in the regulatory decision-making space. It can be early clinical endpoints, surrogates for later endpoints. It could be biomarkers, imaging, whatnot, you name it. We have also heard from David that HTA bodies, they have a very clear preference for long-term outcomes, for patient-relevant outcomes. He had pretty much the same statement on his slides. And it's totally understandable that HTA bodies, they have different types of decisions to make. They have a different context. Their outputs are used for a different step in the process. So it is understandable that different types of endpoints are of interest for the HTA side. However, this may only be part of the story. There may also be difference in the type of uncertainty, the level of uncertainty HTA bodies are willing to take. So you get the impression that HTA bodies tend to be a bit more conservative when it comes to these kind of endpoints and the risks and uncertainties associated with them. So we put here a statement from one of the UNETA 21 guidelines to illustrate that, where they sort of say, yeah, the use of surrogate outcomes is controversial because very often in the first place, the validity of these endpoints has not been robustly established. And that statement has an evidence side to it, I guess, but also a methodology and criteria side. You know, if the hurdle is extremely high for certain disease areas, for certain setups, it may just be very difficult to cross that hurdle, right? So it's methodology, but it's also criteria. What is clear, once a surrogate endpoint has been accepted by major regulators, such as the EMA, such as the FDA, that does create a very strong incentive for the industry, right? So it's very understandable that that will have an implication on how trials are designed and what kind of evidence can be reasonably expected at the time of launch. And it may not be practically very likely, or there may be ethical concerns, etc. And long-term data may just not be a realistic expectation in certain disease areas then. This is not to say that one side is right and the other side is wrong, clearly not. And it's also not to say that there's only one regulatory view and one HTA view. There's also quite some heterogeneity across HTA bodies. So the problem more is that for the industry, this uncertainty or sort of this heterogeneity does create quite some tricky situation. It is a real dilemma. What should we then be doing here? What should be the right evidence packages and what can be reasonably expected? Because if there's too much heterogeneity, if there's this lack of alignment, it may actually have negative implications on the speed to access, on the incentives for certain innovative therapies. Now you may wonder, why are we bringing up this topic again? This is not new at all, but I guess it does get a new level of urgency given the EU HTA regulation, which was very nicely summarized by David, with oncology and ATMPs being the first therapy areas which go through this new EU HTA process. We have exactly therapeutic areas where this type of endpoints are particularly relevant and often used. So with that, I'm not sure what I'm doing with the pointer, but I just, oops, use that. Good. So the questions we put together are summarized here. So first, what is the panel's opinion in terms of pragmatic approaches, pragmatic standards to have to achieve some sort of homogeneity, of common standards when it comes to these questions? I'm Swiss, so we like to talk about pragmatic solutions. So pragmatic meaning that these would be helpful for all stakeholders and viable in practice. If you have a set of thresholds which is essentially impossible to meet in certain disease areas, that may, pragmatically speaking, not be optimal. Then secondly, I think we cannot talk about surrogacy without talking about uncertainty and ways to manage uncertainty. So again, if you have a very strict set of rules, a very high initial hurdle, that is a way to manage uncertainty, but it's not a very dynamic one, right? So there could be other approaches. There could be ideas to more manage uncertainty along the way and have something like a life cycle idea of evidence generation and life cycle HTA decision-making in that sense. always with the objective to create the right incentives for innovation. There's no interest in having decisions based on flawed endpoints, but there's no use in terms of, if you put hurdles which are impossible to cross. The first two questions focus more on the end side of things, but of course, we also should talk about the planning. So what are the things, is this only a planning thing? There is this idea of the parallel joint scientific consultations. Is that the solution to everything? Or actually, are there other questions that will still be there? I'm looking forward to hear the panel's opinions.
Right, well, I guess, thank you. That means me, I suppose, seeing as I'm the sole HTA representative here. You raise a lot of really interesting questions here, and I'm going to. It's kind of hard to answer all of them, but maybe I should. Okay, you mentioned the UNETA guidelines about surrogate endpoints, right? And I think from a HTA perspective, it would be irresponsible not to mention the question of validation, the question of evidence that is available to kind of support surrogate endpoints. But even within those guidelines, there is a recognition that there is a spectrum of evidence that can be used to support surrogate endpoints, from maybe arguments based on biological plausibility alone at one extreme to maybe, IPD meta-regression of multiple RCTs at the other. So, I think in some sense, we have tried as best as possible to be pragmatic about that, I think that HTA agencies in general we're not oblivious to these challenges, right? And I don't think it's, and you're right that we don't want to set, it is not the goal of HTA agencies to set an impossibly high bar that cannot be practically met. I suppose the question is, can we agree maybe on, mutually acceptable endpoints? And of course, when we talk about surrogates in oncology, maybe we talk about progression-free survival or something like that, I mean, there is the question of to what extent does progression-free survival predict overall survival? But there is also the question of, is an improvement in, progression-free survival a benefit in its own right? And this, HTA is not exclusively concerned with using PFS to model OS. So, quality of life benefits are very important there. So, and certainly from, an Irish perspective where where cost effectiveness modeling is a big part of our approach, often PFS is used as a surrogate, not for OS, but actually for quality of life. And that is something that's quite manageable to collect in clinical trials, to have more attention to I suppose, collecting PROs both before and after progression. So, what a surrogate for what, I suppose, is the question here, right? So, I mean, to some sense, I don't think we have algorithmic rules, just to address your first question, right? I mean, certainly, at the European level, the joint clinical assessments, I mean, it's beyond the scope of that to actually prescribe such rules, to express preferences for certain endpoints. It's just, these are factors that have to be taken into consideration. I think many of us already do. So will I go through your three questions? Is that probably the best way? Okay.
Benjamin, maybe you can help, and then we go back when we have heard more. Yeah.
I'm not sure whether I can help, but because I'm not an HTA person, of course, but I would want to take one step back. You have on your previous slide the assumption that regulators accept the surrogates and set precedents, and I'm not sure if this is always the case, especially if I look at your examples, oncology and ATMPs, especially in ATMPs, when we think about gene therapies or other novel therapies, yes, we might accept a surrogate endpoint, like in oncology ATMPs, we might accept objective response rate for licensing, but at the same time request sufficient follow-up data, which is then in the specific obligations. So it's not that your trial ends when you have the label. So you will need to collect this data anyhow, and it's not that we don't want to see that. And we also have a strong time or a difficult time in making these decisions, but there's room to maneuver. So there is possibilities to make decisions also on this preliminary data and surrogate data. But we also want to see patient-relevant data, perhaps from a slightly different perspective than HTAs do, but it's not that we then say, well, we are completely fine with surrogate endpoints and we don't care about it all, at all about more relevant endpoints to the patient. So this might be a slight misconception. And the question then is really, is it always correct to assume that we don't want to see this data or don't need to see this data? And I would say that's not the case.
I'm completely agreeing, but I want to put it somehow more clear. I think our points of view are actually not different. Not different in the sense that we use surrogates, but we demand that there is no detrimental overall survival. Now, in dossier after dossier, it is going back and forth and saying, oh, but the data is not mature. We only do that when it could be mature. If it is clear that it can't be mature, we let go. So it means that, or you say, I stick to OS, but if I see that it is not possible, I back down and I go for surrogates. Or you say, I accept surrogates as long as if we can know OS, we'll ask and we'll push until we have it. So there is actually conceptually not a difference. There is a difference in approach, but there is not a difference in concept. So I think we will be able to harmonize and come to something pragmatic.
Well, I certainly do hope so that this is going to be the case. Indeed, I think there are very different judgments made, right, on these things. You know, whether always is possible or not, I think different stakeholders make very different judgments on that exact question. Or on the meaningfulness on PFS, for example. Again, we do see very different reactions to that question, and some have much more algorithmic rules than others. Also, some systems struggle a lot with modeling approaches, say. And we tend to see that those countries where economic models are being used, for example, they are much more used to assumptions of all sorts of things, and may tend to be more open to certain inputs. So the way these judgments are made are just very different. And this heterogeneity does create, of course, a mess, so to speak, and is not helpful to create the right incentives for companies to design their programs. I guess this lack of clarity is something which can be an issue. And with UNETA, obviously with UHDA, the hope would be that we achieve some sort of harmonization where it is possible.
I think I would short answer case by case. I'm not going to do that. But since there are different approaches and I'm talking oncology, there is a whole lot of different treatments popping up with different specificities. And it is nearly impossible to see upfront what will be reasonable. Therefore, case by case, what can be expected as a risk to OS in case of chemotherapy, in case of other more targeted therapies? Well, it is working from the mechanism that will tell us, yes, we've got to push through, or no, we can leave it alone and go for PFS, and PFS is a real advantage. So it is something that has to evolve. It has to evolve now, but it is not an easy issue, in my point of view. And that is also an interaction between clinicians that have to tell us where we've got to look. And in oncology also, there is a huge problem, because when the targeted therapies will move towards first line, we actually will cure people. And it will be a fraction. Our statistics are not adapted. If I say I cure 1 out of 10 and there are no negative benefits, well, let's jump in the air together. But our statistics are not. If you say, okay, we've got a therapy and now we really cure people, well, I think our statistics work with, well, if it is not too close, the efficiency to 100%, and if it is not too close to 0%, And if there is the proportional hazards more or less functions controlled for, let's be honest, if therapies start to really work, we will not be able to use those statistics. So there is a huge evolution to be made on methodological side, and so also in guidances and in approaches. And I think that variability is there an embryonical start of what is to come.
Question from the audience?
Yeah, or we have a comment from the live as well, yeah.
There's one's coming online. So it says linked to the use of surrogate endpoints such as PFS. Would it be possible to clarify what a non-detrimental effect on OS, how that may translate in the hazard ratio? Thank you.
Who wants to take this question? I'm the chair, I'm sorry. Florian.
So,
okay.
We'll turn Well,
I kind of have a way to avoid the question because I think it's actually partly the wrong question. If you want to see clinical relevance, it's not the hazard ratio alone which will determine where you are. It will actually also be very much related to the absolute death incidence. So one way of dodging the question is if you think in terms of hazard ratio, you will not get an answer.
Could I perhaps return to the questions 2 and 3 you had there? Because I think I actually do have some thoughts on this I'd like to share. So maybe starting with question 2, you mentioned, uncertainty management, life cycle HTA. From a HTA perspective, I think this is something that, we are absolutely interested in. But I guess there are 2 conditions. And I mean, one of them, it's sort of out of our hands. It's really up to payers as well, whether or not, they are willing to say, make therapies available while they await further evidence. And that's kind of out of, that's out of our hands as HTA. But assuming they are, yes, conditional on, this kind of post-launch evidence actually answering the questions that we need answers to. And, sometimes promises to set up a registry and collect overall survival is not really enough, right? I mean, we need well-designed studies with sort of clear criteria for both success and for failure, So yes, we're interested in it, but again, it's this distinction between data and evidence, right? I mean, if done well, we absolutely support it. And, I mean, in the best case scenario, maybe you could run, I don't know, pragmatic RCTs. Maybe that's not going to be possible in all cases, but still you can have, well-designed, non-randomized studies that can give clear answers. So a conditional yes from me on your second question. And as for the third one, you mentioned joint scientific consultation. I mean, I think you say, I'm not listening to what regulators are saying. I don't know if tension is the right word. I'm not sure if we really disagree all that much. Over what's relevant and what constitutes a benefit. I think what joint scientific consultation can do is it can allow industry to plan studies that will answer both sets of questions, right? And it gives you that opportunity to do this early on because I don't think we're actually regulators and HTA are so far apart. And I mean, joint scientific consultation is an opportunity to, I suppose, decide on a study design that can answer both sets of requirements. So, but I certainly don't feel tension here with my regulatory colleagues up on the stage, but maybe they disagree.
Now it's us. We're waiting for a long.
Thank you. My name is Klaus Eckhardt. Formerly regulator, then industry, so knowing kind of both sides, not being a statistician. But one minor addition to the discussion addressing and generating evidence to both sides, surrogate on the one hand, maybe earlier followed by outcomes, also is included in the proposed regulation, which explicitly says for the regulators, if there is substantial uncertainty on surrogates, there needs to be generated further clinical evidence on the respective outcomes. And I think this is kind of the life cycle approach which we have already been discussing. Obviously, I would really also support and agree that we need guidance to when substantial uncertainty is the case in order to have a better planning as early as possible in the place. Thank you.
Thank you. Maybe one quick question, or does somebody want to comment on this? I think we need to move to the last question soon. Sandra, you're fine. Okay. So, and I want to say that this excluding the detriment in OS is a lot written in scientific advices. So I invite that person to submit that question to this session next year. So we can't avoid that question then this time and have proper time to discuss it. Yeah, thank you. So Susie.
So hello, I'm Susie Robson. I'm a statistician in Roche. And my question is definitely the shortest, but I managed to put it over 3 slides. So just to say, I am not a data manager, which you might think when you look at this question. And now it works. Sorry. So sorry, I repeated all. Yeah. So I'm a statistician in Roche, and I have a very short question just before lunch. So let's see. What the regulators think about this. So I'm concerned about basically trials in rare disease, which are really challenging, and often then actually it's the investigators who are in specialized centers really interested in pursuing indications for their patients via an investigator-initiated trial. However, they do struggle to meet the level for data collection and data cleaning, as the trials are sponsored by, compared to trials sponsored by companies. So I think it doesn't work. Okay, so how flexible are regulators with respect to data quality issues from ITTs and particularly sparse data collection or less intensive medical review of data? Regulators willing to accept single-arm trials conducted by investigators with supplemental data from sponsors from already approved indications. So, I mean, so for rare diseases, I mean, where there's not much else, right, for patients. And this is very sparse data. And yeah, and I think they're the experts, the investigators treating.
It's an open and honest question. So who wants to? Andreas, please.
I think this is really an area where it's very difficult to make compromises, because I think data quality is really at the heart of all of our decision-making processes, because when we cannot rely on the data that we have, it's really difficult. I mean, of course, this may depend on the type of data that we have, but here with regard to data quality, and I have also some experience with investigator-initiated trials, and it's really a problem, you're right. But I think I don't see why we can lower the bar just because it's investigator-initiated. And maybe, but isn't it, maybe can I give the back to you? So maybe the industry, they usually don't you have knowledge of these trials that are ongoing, because usually they ask you maybe to provide the drug for the trial and so on. So is the responsibility not more on your side, that you try to help them to improve data quality rather than ask the regulator to make compromises with data quality?
But if the sponsor is the investigator.
Now it's
Okay, sorry. The investigator is the sponsor, and yes, it's actually the investigator interested, right? They want to care for their patients, and it is just the standard in pharma industry. I mean, we have all this machinery going on, right? That's not at the hospital, and that's clear. It's a different setting, and it is very. You always have to it's a collaboration. Then it can be really very difficult. And I just wonder if one couldn't open a bit the door for these patients so they can have a treatment, available, right, in these settings.
So just to Andre's point, I think, it's not possible to compromise on the quality of the data, irrespective whether it is investigator-led or not. I think we require that all trial data generated is robust and meet the standard GCP requirement. In terms of the setting of where you have an unmet need, etc., that's obviously where there is clearly an unmet need that we may not reach the level of evidence that we would expect, but the evidence always has to be good enough for the benefit risk assessment, taking into consideration that it is an unmet need. So it's not, it's a case by case again, basis, because where there is clearly an unmet need and there is clearly no option for getting more evidence, then obviously that is considered in the context of the totality of the data, not just the primary endpoint.
Lucas, please, or Andra.
Maybe I can also add that I would also agree completely with Andreas. And I think we have to realize that if we are looking at a formal, like an application, it is still a sponsored application, and the data, the sponsor of the application, in my view, still has responsibility for the data quality. And we cannot sort of, from a regulatory perspective, look at sort of two tiers of approval or authorization. We authorize on a regulatory basis, and the regulatory application needs to be supported with the data that is consistent with expectations for a regulatory application. And I think, for many of these indications, the investigators are working with authorized products for some other indication. So it's not like the authorization or rejection of a variation application is depriving patients, of that product, because in many countries, most countries, physicians still have the possibility to, prescribe off-label in that sense. And as Kit pointed out, they end up in treatment guidelines on many occasions. So it's not like, the rejection of that regulatory application is depriving anyone of anything. It could be, depriving them of reimbursement, but on the other hand, I think, we still need to work on one sort of set of expectations in terms of data quality. Thanks.
Perhaps just to add one more complexity of these trials, we sometimes see them and we have to assess them. I have to admit this as well, but it's also a question of data ownership. So when you are not the sponsor, we often case here, well, we don't have access to the raw data, we cannot answer these questions and all that. So this is really complicated procedures. And it's really problematic. So I think there needs to be a good agreement also between the investigators and the companies to be able to have access to the data and to allow to improve the quality of the trial, to allow to also answer questions of regulators. And as I said, we need to, whenever you file these procedures, well, we need to assess them, but I can tell you it will be difficult for both sides. And we need to find a way to maneuver through this, to try to address the data quality issues, also other issues of the data which was accrued. Also the second question on single arm trials, well, this further complicates things, but I don't want to touch upon this, and we have a session on these kinds tomorrow, I guess. I think that there's not a general rule that we can lower the bar just because it's an investigator-initiated trial and just because it's a rare disease. Of course, we will take it into consideration, but there's no general rule that we lower the quality standards by, I don't know, a specific percentage and then everything's fine. The discussions will be really hard and you need to be prepared for that.
Lucas, please.
Yeah, I don't know whether I will be able to put this into better words than the others who already made basically a very similar comment. So I think as a basic principle, we really don't like lowering standards in study design or data quality, also in rare diseases. It's clear that we will have limited information because the population is small. And then in the end, we will have to weigh all the benefits and the risks which are inherent also to lack of information and inherent to maybe poor data quality. We have to weigh all this together, and that's very difficult. So if the data quality is poor, then this can be really critical, even if we understand why that may happen. On the other hand, we will take anything that we get. So if there is any information out there, if there's any trial that has been conducted, even if it has problems, we will take it into consideration, and I think we have to. The other point that I wanted to make is very short, maybe, and it's related to the second question. I think we can give a very brief answer that we have a guideline on clinical trials in small populations that clearly states that randomized trials, even if underpowered, are more valuable to us as regulators in small populations. So that's the starting point for the discussion, and any deviation should be justified, I guess.
I think just to the point that Luca made about really making a clear distinction about limited data and data of bad quality, because they are two different things. You can still have limited data, but it is of good quality. Mm-hmm. And with that, I think as regulators, we can live with that. And as Luca said, there is a guidance with some recommendation on how to optimize the design, etc. But you know, the starting point is the quality, and if you cannot get that past that, then there is really nothing for us as regulator to assess if you cannot provide assurance of the data quality.
I mean, if it's like not a validated system where they record, or if the CRF is not capturing everything that we would normally capture in a company-sponsored study. So I'm not saying it's poor quality data. I'm talking about lowering the bar to adjust a bit for the trials they've done at sites.
There is something that Kadisha said earlier, but he said it very fast. The study has to be GCP compliant. Like, but really, like, there is no, like, you know, we can't relax certain requirements. So for example, the randomization system has to be GCP compliant if there was one, or the data collection system has to be GCP compliant.
Yeah. It's kind of a bridge to the point I would like to make. We also investigator-initiated trials fall under the same legal framework. So exactly the same in terms of data quality is actually expected. And I'm a little bit more optimistic to the extent that it's how that's implemented, because I think they will typically also have to write protocols, run them by METC committees. They will have CRFs in place, so it might not be up to the standards that companies have currently, but I think it's not such a, let's say, bleak picture that we have to assume bad quality because it's investigator initiated. In terms of completeness of the data, I also a bit of a personal picture on the model of pharma and CROs and the level of detail and oversight. Not all of that contributes, let's say, proportionally to increased quality. Having less data, but very well captured to people who actually know what they're treating, might be very good quality, maybe a bit less than we are used to, but it might be very high quality data which you can rely on. So I would like to make the discussion on data quality also quite precise. So what's data quality? Or are you missing stuff that you generally do? And that's the extensive stuff on concomitant medication, all AEs, which you will never see in investigator-initiated trials. You will see much more targeted investigation and safety than we're used to in company trials. Now, quite a bit of that we can live with as long as what is there is of high quality. So I will nuance that a bit. I have a very brief answer to the second one. Probably we have accepted it, but it's not what I would design for. That's the.
Any comments from the audience? Nafsika, please.
Thank you. Nafsika Kronidu from Roche. Throughout the day, I heard the word pragmatic several times.
Just close.
I heard the word pragmatic several times, and I was actually very happy to hear the comments at the end towards less data, but high quality. And so there is, of course, this concept of randomized pragmatic trials being discussed in several different forums. And I was interested to hear your thoughts about how much data variables is enough from your perspective and how you see the prospect of randomized pragmatic trials in Europe.
So it's quite a different, about randomized pragmatic trials, kind of a bridge, but I think it links to the fact that less data, but good quality. So how much less is still acceptable? It depends. Please. Tommy? No?
I lower the bar for any remaining comments. I have not much to say, not much experience. So I hand over to my more experienced colleagues.
It's not because I've got gray hair that I'm experienced. By the way, no, I think that we've got to go with pragmatic trials to something that resembles a natural patient flow. We do randomized controlled trials to actually capture things that we can't measure. Therefore, somebody can react extremely good in being in the control, and somebody else extremely good being, and it can be on completely different reasons, because we are complex human beings in complex environments. So we use randomization to try to isolate causality to a certain uncertainty. So your answer can't be seen separately from our data collection process and our sampling process. So if you say, I need nearly no variables, if I've got a huge sample of trial sites, and within those trial sites, I've got a good capturing of patients, then I'll be a representative, and I don't need to measure. I only need to measure the output, the end variable. I even don't need to stratify. Stratification is something we add because we are sure we can't do that right now. So with pragmatical trials, it will be a trade-off, and sampling will become extremely important. But it will also be able to use also statistics to capture variability between circumstances, trial sites, and that kind of things. So it might, if well implemented, open a whole new world. But the answer, what variables do we have to measure at present? I don't think there is scientific evidence to answer your question. We'll have to build it up and see what is possible and see what is reasonable.
You have one question or a comment from the line?
Yeah, so someone online has asked, how often are pharma companies actually submitting data to health authorities, which come from sort of IITs that we're talking about? what's your experience of the actual quality of the data that's submitted? And so, yeah, how often is that meeting your expectations of data quality? So.
A frequency on that level, I didn't do the statistics, so I'll pass on to.
Yeah, only a gut feeling. I've seen a few and with very diverse data quality and diverse size. So very big studies with collecting data on hard endpoints that left a few questions open because they were pragmatic and did not collect all the data. So there were uncertainties because of that we needed to weigh as usual. But I've also seen with rare diseases, especially trials, very good trials, IITs, but also some that really had problems with data quality. And then the resolving process was complicated, including inspections and lots of uncertainties. So I guess it's a range.
From my experiences, I've seen some very good applications and also not so good. And the good application tend to address the data limitation by augmenting the data. And that's all generally been quite helpful in the benefit risk assessment. But overall, I think I've seen both. It's very variable. But that's also to say that even with the sponsor-led clinical trials, we have also seen very bad data. So, so I think it's not so much who is running the trial, whether it's a sponsor or investigator, but actually the scale and expertise and experience of the people involved in the trial. So that's just minor comments.
Thank you. So I really want to thank everybody who submitted the questions and presented them. And really, the regulators, it's not easy to sit there for 2 hours and answer this kind of varied questions. So thank you, everybody.
I would like to state, as an industry chair of this session, we should really acknowledge and cherish the openness and the willingness of our regulatory colleagues to answer these questions here on the spot. I think this is something really special and deserves maybe yet another applause. And now up for lunch, and we see you back in 2 hours.
Let's put it on.